So madam is talking to you, I think. Then we start. Okay. Good evening, one and all. Shri Kumarswami Mahavidyalaya Ausa organized a very long series, that is world record series. Today is the 109th day of the series, and the keynote speaker for today's session is Dr. B Vishnupriya Madam. So first of all, I welcome to you, Madam, for the dais. Then I welcome to the chief organizer of the World Record Series 2, Honorable Principal M. Ambedkar, sir. I welcome to all my colleagues from my college. IQAC coordinator Dr. Sanjay Kale, sir, Dr. Sudhir Garad, sir, Dr. K. N. Kadam, sir, Dr. Karar, sir, Dr. Anita Dhole, madam. Then the vocational supervisor Praveen Woodke, sir. And lastly, I warmly welcome to all those participants who joins regularly to the series and motivate us. So welcome to you all. For today's session, the topic is basic of biotechnology and its application. Now this topic is going to present Dr. B Vishnupriya Madam. Before the presentation, to start the presentation in short, I would like to introduce today's keynote speaker. The speaker is Dr. B Vishnupriya Madam. She is an assistant professor from the Department of Biotechnology and the college Kongunadu Arts and Science College from Coimbatore, Tamilnadu. So with this short introduction, now I would like to request the keynote speaker, Dr. Vishnupriya Madam. Please start your presentation on the topic Basic of Biotechnology and its application. Welcome, Madam, please.
So thank you so much. Thank you for your introduction. It's my great pleasure to thank you, sir. It's my pleasure to share about biotechnology in this great occasion. And I express my sincere thanks to our principal sir and also and also the college for inviting me for today's session. It's my very honor to privilege to present my topic here. Thank you so much, sir. Okay, I'll share my slides, sir. Sir, can you see my slides? Is my slide visible? Yes, sir.
Yes, madam. It's visible now. It's
Okay.
okay.
Yes.
Yes, madam. Please.
So topic about today's presentation is about basics of biotechnology and its Department of Biotechnology, Kongunadu Arts and Science College, Coimbatore. So the first of all about biotechnology. We can see that is biotech living organism to obtain the useful products. It's nothing. So in this line, I explained that's produced by Bharat Biotech and Dr. Reddy's Laboratory, India. This is a great achievement and it is a technology, Covaxin and Covishield discovery. The development of biotechnology. He even also produced, even the wine is also produced with the help of Saccharomyces cerevisiae, and it is an important organism which is involved in the production of wine, and that is by using the grape juice, the organisms are inoculated, and by the absence of oxygen, the wine is produced. So this is an important achievement, the fermentation technology. Coming to the discoveries and advancements in biotechnology, so, important advancement in biotechnology. I try to explain about the basics of cell. So, in this, a human, it is a comprises of different internal organs. Each organs are formed by the group of tissues. So, these tissues are formed from the millions of cells. Okay. So, and what are the components which is present inside the cell? Which means inside the cell, we are having the nucleus. Inside the nucleus, we have the chromosomes. In the human, we are having 46 number of chromosomes. 46 means 23 pairs of chromosomes. Here, the 22 pairs are autosomes and one pair is allosomes. So, in this, it is about, I'll just give a small introduction about deoxyribonucleic acid. It is a double helix structure. In this double helix structure, there are four important base pairs are there: adenine, guanine, cytosine, and thymine. This important base pairs are that is importantly coding a gene, which is coding a protein. So this activities, all the day-to-day activities, and also it conducts the genetic information or transfer the genetic information from one generation to the next generation. So this is the important discovery of Watson and Crick, and it is a basic it is important genetic information which transfer the information from one generation to another generation. So next one is the types, different types of biotechnologies are there. So here we are. I'm not going to discuss all the important types of biotechnology. So I just importantly cover about the animal biotechnology, plant, and the microbial biotechnology. But there are different streams in the biotechnology. So coming to the animal biotechnology. So in this animal biotechnology, we are what we are doing in this animal biotechnology is we are genetically engineering the organism, or we are genetically modifying the organism or the animal, and in order to improve their suitability for agriculture, industry, and also the pharmaceutical application. We are we are modifying the genetic sequence of the wild type animal, and we are genetically modifying the animals into us for our needs. So that is we are doing in the animal biotechnology and also through recombinant DNA technology. So let's see what are the important things we have achieved in animal biotechnology. Yes, this is the thing. We may hear the term that is cloning. So this is the cloning means nothing. That is the exact copy of the organism that we can produce. In the that is the term is called cloning. Here in the year 1997, the first cloned animal Dolly, the first cloned animal Dolly is produced through biotechnology, which is called recombinant DNA technology. Here, what they have done means that is a two sheep they have chosen. One is Scottish black-faced sheep, and another one is pink Dorset sheep. So they have taken two cells from this two animal, and they have cloned these two cells, which means one the egg is taken from the Finn Dorset sheep, and another sheep they have taken they have taken the nucleus. This two genetic material has been fused, and under the in vitro condition. In vitro means outside the body, outside our body, which means in the environment. They have in the plate they have cultured the cells up to 6 days. Once the cells has been grown, this cells has been carefully transferred into the surrogative mother, transferred into the surrogative mother through this catheter. And the first cloned animal is produced in the year 1997. The name is This is an important achievement in the biotechnology year 1997. In the next slide, you may see the genetically engineered animals. That is that is two different animals you may see. First one is the fish. That is called genetically engineered fish. Here, both it is you may see two fish together. Both are 18 months old. The upper one is genetically engineered, and the lower one is wild type variety. So you can see the difference. That is the length of the fish. The wild type fish is a smaller, and genetically engineered fish is larger in size. The purpose of genetically modified fish is to enhance the quality, growth, and also the productivity. And also it is climatic it can grow in the under the climatic stress also. So this is the first human consumption food which is approved by FDA, Food and Drug Administration US, and it has come for human consumption. So in the in this beside you can see the next two animal, that is the mouse. Two mouse you may see. The left one is wild type and affected hair growth, and the next one is the genetically modified mouse. You can see the full hair growth throughout the body. So the mouse is genetic. That is the gene is mutated. The gene which affects the hair growth is mutated through biotechnology, and the complete hair growth you may find in the genetically engineered mouse. So this we can achieve through biotechnology only, through recombinant DNA technology. So by this way we can mutate the gene which is responsible for disease causing and also some hereditary disease causing gene that can be mutated through biotechnology. So in the next slide, you can see the pig, that is the GM pigs, which can say genetically modified pig. So nowadays, people are dying due to organ failure, and also people unable to get the organs on time. So to overcome this problem, genetically modified pigs has been created through biotechnology. So in this, why they have chosen the pig means pigs are very similar to humans in size, organ size, and also the physiology. It is very much closer to animal, and also the genetic similarity also very close to human. That's why the scientists prefer the pig for organ donor. So this type of organ donation is called xenograft because it is entirely different from the human. So that is called xenografting. We are taking the organ from the animal. This type of grafting is called xenograft. Okay. So here in this image you can see the first person in the world to get a heart transplant from the genetically modified pig. They have successfully transferred the heart from the pig, that is genetically modified pig, to the person, that is David. The person's name is David, and the surgeon who successfully done this surgery, that is Bartley Pig Griffith in University of Maryland School of Medicine. So uh, already I said pigs are the ideal donors because of their physiological metabolism and also the immune system, which are more similar to the human being. This is the successful organ transfer from the pig to human. And also, there are different, as already I said, there are different domains in the biotechnology. So, through this biotechnology, we achieve many things. And one of the important achievements is embryo transfer, which means you may hear IVF, in vitro fertilization. So, people who may unable to give normal birth, they can go for in vitro fertilization. That is, the fertility center is raising nowadays, and also this fertility center, what the people are doing means they are artificially transfer that is the fertilized egg into the nutrient wall of the to the healthy baby. This can be achieved through the biotransfer technology. Okay. So the next is plant parasites. So what I explained in the animal biotech so that the different applications. Here the plants are involving in the different different applications. Here we are modifying the genetic traits of the animal through by genome editing, and they are importantly used for agriculture and also the economic growth. So very first, you may hear the golden rice. This golden rice is genetically modified to produce beta carotene. So beta carotene, it is a precursor of vitamin A. So vitamin A is a fat soluble vitamin, and it is a so vitamin A. When we consuming the vitamin A rich food, we are not consuming the form of vitamin A. It is in the form of beta carotene. That is the precursor form of the vitamin A. So this vitamin A. So in this rice, you may see the difference. The left side it is white in color, and it is next one is yellow in color. This yellow in color is a golden rice, which is vitamin A enriched. So this can be achieved through biotechnology. So we need vitamin A for healthy skin, immune system, and also vision. So the next one is the BT cotton. BT cotton, that is, a cotton is an important product for the economic growth of our country. So once upon a time, the cotton growth, the cotton plant is completely abolished, or it is the production has been completely lost due to the worms and also the pest. So to overcome this problem through the genetic engineering, BT cotton, that is a Bacillus thuringiensis cotton, is produced, which means a gene which is taken from the Bacillus thuringiensis, that is a bacterium, and it is incorporated into the cotton plant. So the plant normally, that is naturally, produce the natural insecticide which fight against the pest or the germs. So now the BT cotton, this is the left side is the BT cotton, and the right side is the non-BT cotton. You may see the difference. And in the lower, in this image, you can see the good cotton variety through the biotechnology. So the benefits of this BT cotton is the yield of cotton is increased due to effective control of worms, and also the reduction in insecticide using the cultivation of the BT cotton, and also the cost of cultivation also reduced through the biotechnology. So the next one is the BT brinjal. BT brinjal is not Bacillus thuringiensis brinjal. So you may hear this BT brinjal has been developed to give resistance against the insects. So the left side you may see the wild type, and the right side it is the genetically modified brinjal. This genetically modified brinjal underwent a trial for 7 years in the greenhouse trials and in the location of Bangladesh, and it is successfully cultivated in the region of Bangladesh. And what are the special feature of this BT brinjal is it is natural insect resistant, and also it increases the yield, more cost effective, and has a minimal environmental impact. So, and also another important benefit is this BT brinjal would reduce the pesticide usage in the cultivation by 80%. So on the left side, that is the brinjal is completely affected by germs, affected by insects, and in the right side, that is a fresh brinjal you can see. So the next one is the flavor saver tomato. In this flavor saver tomato, it is the first genetically engineered crop product to be commercialized. So what is the special feature of this flavor saver tomato is normally the tomato problem is the fastest ripening, and also before it reaches the home, the post harvesting loss is much higher. So this can be overcome by through biotechnology. So what they have done is they are blocked the gene which is responsible for ripening. So fruit ripening gene has been mutated. So the flavor saver tomato is is produced by blocking the gene which is responsible for ripening. So in this process, the post-harvest loss is completely minimized, and also we can get fresh and improved flavor of the tomato, and we can reduce the spoilage of the tomato also. So next is the plant and animal tissue culture. When coming to the plant and animal tissue culture, so in the slide you can see this plant tissue culture. We can grow a plant. We can grow a plant in a small bottle. In a small plant tissue culture bottle, we can grow a plant. So how we can grow the plant? Which means we are taking a part of the plant that is called explant. We are taking explant and we can grow a plant inside the laboratory. And this inside the laboratory, we can grow any type of plant. The plants which are having the drought resistant, that is a biotic and abiotic stress tolerant plants that we can grow inside the lab. And also once a root and shoot has been developed and it is transferred into the uh, uh, greenhouse, which means that is a primary hardening. So we are not directly taken to the outside environment suddenly. So we slowly taking the plants from the lab to outside environment. So in this here you may see the complete overview of plant tissue culture. So from the mother plant, we are taking a small part from the mother plant. So and we can grow in the bottle the plants that obtain as height, and it is transferred into the primary hardening. And once it grows well, and it grow and it take and it is it takes in for the outside environment, that is in the outside environmental condition. So the plants that what any type of plant that we can cultivate it in the plant tissue culture, which means endangered plant variety we can protect through plant tissue culture. So, as same in the animal tissue culture, from a small cell or from a small that is a tissue, uh, we can we can get a full organ through animal tissue culture. So, this is the biosafety cabinet we are using. A small cell which is taken from the human, and we can grow a complete cells, and we can preserve it for future use. This is animal tissue culture. And the next one is a very interesting one that is called organ printing. This organ printing, we can, so already explained that is from genetically modified pig, we get organ. So now here, that is organ printing is we can print our organs through 3D printing technology. So we are filling the bio ink, that is the ink is filled with completely our cells. And the cells are fed with the nutrients that is called the media, and the mixture is loaded on the printing chamber and the by layer. So the finished product, that is a human ear, is developed under in vitro condition through this bio ink. So this artificial, that is organ printing, is also in the it is gaining much more interest, and for that is much more interest nowadays. Through bioink, we can produce a full organ. Any type of organ we can produce through organ printing. So coming to the last session, that is environmental biotechnology. In this environmental biotechnology, that's a lots of pollution, oil spills that can be easily recovered by bioremediation technique. That is called bioremediation technique, which refers to the use of living organisms to remove the contamination, pollution from the unwanted substance, soil, water. So here the image that is I'm showing here Professor Anand Mohan Chakrabarti. He is a superhero. He is the discovery of he discovered a super bug. A super bug name is called Pseudomonas, which is a bacteria. It is the first ever bacteria which is patented. First bacteria which is patented by the Indian. scientist, that is Anand Mohan Chakrabarti. What is the speciality of this bacteria is this bacteria eats oil. So, is there any oil spills over the ocean? In the next slide, I'm showing that oil-eating bacteria. Is there any accidental oil spills which covers the entire ocean? It kills all the important creatures which is present in the marine. So this can be easily recovered, and this can be easily recovered by the through Pseudomonas bacteria. So in this first image, the oil is entirely spilled, that is covered entire ocean. In the second one, the powder form of the superbug, that is Pseudomonas bacteria, is sprayed entirely the oil spilled area. So within 5 to 7 days, the entire oil is eaten by this bacteria, this oil eating bacteria, and it becomes eco-friendly. So it is the entire oil spills can be recovered by using this bacteria. So for this genetically engineered bacteria, the Mr. Dr. Anand Mohan Chakrabarti um, filed a patent for this bacteria. So this is oil eating bacteria, and we can say this is superbug. So coming to the marine biotechnology, in this marine biotechnology, it is emerging field, and it has a significant potential for developing new products and also creating healthier, stronger, and more sustainable food products. So it is an important access. It is a pleasure area and also access research and development in the many exciting possibilities for the future. So what is the importance of the marine biotechnology is the transgenic glowfish. So transgenic glowfish means for the this type of fish are bred. It is modified into transgenic fish, and we can produce different colors of fish through recombinant DNA technology like fluorescent green and the cosmic blue. We can produce different types of ornamental fish through transgene or the micro ejection technique. We can produce different type of fish, and it is for ornamental purpose or ornamental fish for trade. So coming to the last title, that is the important advantages of biotechnology. So this biotechnology offers a variety of advantages, solutions to the critical problems. Some of the important things are for the human welfare only, the biotechnologies are playing an important role. So creating healthier, stronger, and more sustainable food products to boost our nutrition in all the food products, and also treating many diseases, especially hereditary diseases. Those diseases do not have cure. Everything and all we can cure. We can cure by altering the gene or mutating the gene, and we can prevent we can prevent the disease to spread from one generation to next generation by mutating or knocking out the gene. And also we can designing the medicine to boost the health and the longevity of the people, animals, and the plants. And finally, in the plant biotechnology, we can produce n number of plants, and also we can protect endangered plants, and we also improving the crop yields, and we can get more benefits through plant biotechnology. So, and thank you for your patient listening. I hope I Thank you, sir. And I hope you understood basics about the biotechnology, and you may get a idea about biotechnology and its important applications, sir. So thank you so much. Your doubts. And I just explain the basics about biotechnology and what are all the important domains and streams in the biotechnology. So it is much more interesting domain. And if you have any doubts or anything to ask, please ask me. Yes, I'm very pleasure to share my share it.
Very informative session. I would like to know any
Good
particular
evening.
journals in this area so that we can search for it, the recent ongoing research.
Yes, ma'am, sure. That is a n number of research is going on in biotechnology. That is, you can search. That is a Springer, Elsevier. These are all the pioneered in the publications, and also many books are available online. So you may refer any type of books.
Just
Thank you. The principal sir asked